Welcome to The Explainer. I am so thrilled you're here today. Let's just dive right in because we're about to explore a biological mystery that basically shatters some of the absolute oldest rules of science. Now, we're talking about proteins, but not the rigid, predictable ones you might be used to. No, we're looking at real-life, shape-shifting molecular machines. And, of course, the incredible bioinformatics tools we actually use to track them down. So, I want to start by throwing a question out there that might just challenge everything you learned in high school biology. What if the most important machines inside our bodies are, well, actually missing a few parts? For decades, right, the standard scientific model told us that a protein has to fold into this perfectly rigid, highly structured lock and key shape to do its job. But what if that's literally only half the story? What if some proteins are completely floppy, dynamic, and honestly essentially unstructured, yet they're absolutely crucial to keeping us alive? Here's our roadmap for the explainer. First, decoding protein bioinformatics. Second, the disordered protein mystery. Third, extended versus collapsed disorder. Fourth, shape-shifting proteins in action. Fifth, predicting disorder with PONDR. And finally, modeling structure with Phyre2. Let's get to it.
All right, Section 1, Decoding Protein Bioinformatics. Before we get into the rule breakers, we really need to understand why scientists spend so much time obsessing over predicting protein structures in the first place. The short answer? Understanding biological function at the molecular level is incredibly high stakes. Predicting a protein structure helps us identify specific targets so we can design new therapeutic drugs. It reveals evolutionary relationships, it helps us annotate unknown proteins, and it's basically a direct guide for synthetic biology and genetic engineering. But crucially, it lets us predict disease-related mutations. If you want to cure a disease or maybe engineer a totally new biological function, mapping that molecular blueprint, that is step 1. Moving on to Section 2, the disordered protein mystery. The missing structure.
So for a long time, we honestly thought incomplete folding was just a mistake, an error. But then scientists discovered what we call disordered regions, or DRs. And the absolutely crucial point here is that these are areas of a protein that completely lack a fixed, stable 3D tertiary structure. And just to be super clear, this disorder is happening right in the protein's primary backbone, not just the side chains. The amino acid sequence itself locally codes for this dynamic, fluctuating state. The structural angles are rapidly changing, it's just totally dynamic.
Think about the contrast for a second. On one hand, you have classic, tightly folded structured proteins, the rigid lock and key model we all know. But on the other hand, you have intrinsically disordered proteins and regions. Instead of being tightly packed, they behave more like loose, flexible strands of molecular spaghetti. They completely defy the traditional rules because they function precisely because they're unstructured and flexible. It's wild.
All right, Section 3, Classes of Disorder, Extended versus Collapsed. Let's look at how the mechanics of this disorder actually break down into two main types. First, we have extended disorder. Think of this as, well, random and coil-like. The backbone is highly extended, almost like a beta sheet, but without any of those stabilizing long-range contacts holding it together. The structural angles are just rapidly and constantly fluctuating. Now compare that to collapsed disorder. This one is slightly more organized. Scientists often describe it as molten globule-like. While it still lacks a stable overall 3D structure, it might actually contain some localized, folded secondary structure hidden inside that globule.
Next up, Section 4, Proteins in Action, Shape-Shifting Wonders.
Okay, here's an incredible real-world example of extended disorder. We're talking about calcineurin. It's a phosphatase that plays a massive role in activating your T cells. Within its structure, there is a huge 95 residue disordered stretch. It is totally floppy. But here's the kicker. This floppy region is actually an active trap. It houses an autoinhibitory peptide and a super crucial binding helix.
So, what happens when it actually goes to work? We get this dramatic event known as a disorder to order transition. When another protein called calmodulin binds to that floppy region, it wraps around the binding helix and literally forces it to snap into a rigid, ordered structure. That rapid transition from complete chaos to perfect order is exactly what activates the calcineurin protein. It literally shapeshifts to turn itself on. How cool is that?
Now, for an example of collapse disorder, researchers often point to a protein called clusterin, which handles everything from lipid transport to your body's injury response. Because disordered regions are so loose and exposed, they're basically sitting ducks. They are really easy targets. An enzyme, like trypsin, can easily chop up the protein right at those predicted disordered regions. In fact, if the protein gets artificially denatured, basically unfolded even more, an even larger number of these floppy, exposed sites pop up for the enzyme to cut. You might be wondering, why on earth would evolution favor proteins that don't even have a fixed shape? Well, here's the evolutionary why. These shape-shifting abilities make proteins incredibly versatile. A flexible protein can mold itself perfectly for precise DNA, RNA, or protein recognition. It allows the protein to modulate its binding specificity and affinity, acting kind of like a molecular universal adapter. It enables molecular threading, and, as we just saw, it allows proteins to be activated by cleavage or binding. The flexibility isn't a bug, it's absolutely a feature. Section 5. Predicting disorder with POND. Finding the chaos. Now we're getting into the bioinformatics tools. PONDR stands for the Predictor of Natural Disordered Regions. It's a highly specialized tool that relies entirely on sequence data alone. And the workflow? Elegantly simple. Step 1. Researchers just input the raw amino acid sequence. Step 2. PONDR processes that sequence using complex machine learning algorithms, specifically models known as VLXT and VL3. And step 3, it spits out a probability score for every single amino acid or residue in that sequence. That score ranges from 0 to 1, basically estimating exactly how likely that specific spot is to be disordered. And when you run this, the output takes all that abstract theory and turns it into hard data. The system sets a crucial probability threshold right at 0.5. Anytime the algorithm's scores spike above that 0.5 mark, it's basically raising a red flag, saying that specific segment of the protein is highly likely to be intrinsically disordered. It creates a complete statistical map of the protein's flexibility, literally pinpointing the exact coordinates of that molecular spaghetti. Bringing us to Section 6, Modeling Structure with Phyre2, Mapping Rigid Parts. Phyre2 is the protein homology recognition engine, and let me tell you, it is a web-based powerhouse for 3D structure prediction. Just like PONDR, the workflow here is super structured. Step 1, you input the sequence, usually in a FASTA format. Step 2, the engine uses what are called hidden Markov models to comb through massive databases, aligning your sequence with known protein structures. Then Step 3, it outputs a highly accurate predicted 3D model of your structured protein, completely paired with a confidence score so you know exactly how reliable that model is. Phyre2 is just packed with features that are essential for modern research. I mean, it absolutely excels at fold recognition and secondary structure prediction. But what's highly relevant for fields like pharmacology is that it also predicts ligand binding sites. That means it shows you exactly where a therapeutic drug might attach to the protein. And honestly, it grades its own homework by giving you those model quality scores, which totally empowers researchers who are doing things like mutagenesis and protein engineering. So, the biggest takeaway from this entire explainer is how these distinct tools actually come together. We don't just rely on one or the other. Researchers combine them to get the complete picture. You use Phyre2 to generate high-competence 3D maps of the rigid, structured domains of a protein. And then, you pair that with POND to identify and annotate the flexible, unstructured, shape-shifting regions. Together, they allow us to see the protein in its entirety, exactly as it exists in nature. Which really leaves us with a final, pretty profound question to think about. If bioinformatics tools like PONDR and Phyre2 allow us to accurately track and map these molecular shapeshifters, how might manipulating these flexible regions be the key to the next massive breakthrough in biotechnology, or even targeted drug discovery? The rules of structural biology are clearly changing, and the dynamic chaos of disordered proteins might just be where the entire future of medicine is hiding. Thank you so much for joining me for this explainer. Keep learning, stay curious, and I'll catch you next time.